Processing abdominal and thoracic fluid specimens consists of measuring protein in specific gravity, performing cell counts, and preparing and staining slides for cytologic evaluation. Here we will demonstrate how to prepare slides and measure the protein in specific gravity. For most fluids, 3 types of preparations are made: direct smears, concentrated direct smears, and cytocentrifuge preparations. Note that cytocentrifuge preparations require a special instrument and are typically only made in larger laboratories. When processing fluids, pre-label your slides with accession numbers, lab numbers, and fluid volumes or preparation type using a number 3 pencil. First, we will demonstrate how to prepare a cytocentrifuge preparation. Take out a bullet tube, cytocentrifuge cassettes, and cytocentrifuge funnels. Insert the slides and funnels into each cassette. Put on gloves and place a splash shield over your work area. 10 microliters of bovine albumin is placed in each cytofunnel to help the specimens stick to the slides. After the sample has been analyzed for white cell count, make sure the specimen is well mixed. Transfer 1 fluid at a time to decrease error possibilities. Here, fluid is transferred to the cytocentrifuge funnels in 25 microliter, 50 microliter, and 100 microliter volumes. The cassettes are then loaded into the centrifuge and spun at 750 RPM for 5 minutes. Next, prepare 2 direct smears with your well-mixed fluid sample. There are 2 different techniques for preparing direct smears. The first is similar to making a blood smear. Place a drop of fluid on a slide. Then place the clean edge of a second slide next to the drop of fluid. Pull the edge of the slide into the drop and then push the slide forward to create a smear with cells spread out on the slide. The second technique is sometimes referred to as the squash technique. Place a drop on another slide. Then, place a clean slide directly on top of the slide. Pull the 2 slides apart smoothly without twisting them. This method results in a smear that leaves cells less disrupted. When the centrifuge has finished spinning, remove the cytocentrifuge cassettes. Take the slides from the cassettes and etch the area around the cytocentrifuge cells with a dry point marker. Lift the cytofunnel directly off the specimen to avoid disturbing the cells on the slides. Allow the slides to air dry. To make concentrated direct slides, first place a small volume of well-mixed fluid in a bullet tube. Then place the bullet tube in a centrifuge. Spin the sample for only a few seconds to avoid disrupting the cell structure. Note that the sample can also be spun down like urine sediment to make a concentrated specimen. Specific gravity and protein are evaluated from the supernatant in the bullet tube once it is finished spinning. After removing the bullet tube from the centrifuge, separate the supernatant from the settled cells in the bottom of the tube. Then, place a few drops of the supernatant fluid in the well of the refractometer. Point the refractometer at a direct light source to obtain a specific gravity reading. Record the results to the patient's lab sheet and clean the refractometer with a dry Kim wipe. With the lid on the bullet tube, resuspend the settled cells by flicking the tube with your finger until the cells are homogeneous. Draw fluid from the tube with a microhemocrit tube. Transfer drops from the microhemocrit tube onto 2 slides. Then prepare 1 smear using the pull-push technique and a second smear using the squash technique. Allow the slides to air dry before placing them on a slide stainer or hand stain them using quick stain solutions. Once the slides are finished on the slide stainer, they are ready for evaluation.